Hello and welcome back to Science on Trial and Error. I'm Kasia Kuzmicz-Kowalska and I'm the host of this podcast. Thank you for joining me for another episode. I'm so excited that it's already the fifth one. Before we begin the interview, I'd like to say thank you to all of you who follow the podcast channels on different platforms. I would be very grateful if you could share the episodes with your friends and colleagues and help spread the news. Today my guest is Sikora. Matt comes from Poland. He's a biophysicist and a postdoc with a dual affiliation at Max Planck Institute for Biophysics in Frankfurt and at the University of Vienna. He currently investigates an extremely timely topic. He uses simulations to gain insights into the spike protein of SARS-CoV-2 virus. This work is important for both understanding the virus infectivity and the development of new strategies and vaccines. And believe me, Matt explains it in layman's terms. Prior to his current position, Matt obtained his PhD degree in biophysics from Institute of Physics of Polish Academy of Sciences in Warsaw. And then he became a postdoc in the group of C.P. Heisenberg at IST Austria. I've gotten to know Matt through his wife Anya, and they are both amazing. Matt's scientific resume is most impressive, and his path has been in some sense unusual, with theory and experimental work getting more and more intertwined with every step. Now he plans to start his own group, and I'm certain that he will succeed. I'm very happy that Matt joined me to tell his story, discuss the importance of work-life balance, and share his perspective on how to improve academia. Regardless of your current career stage, I think you will find our conversation useful when looking for your own way in science. Enjoy, and please welcome Matt Sikora. Hi, Matt. Welcome to my podcast. Thank you for accepting the invitation.
Hello, thank you for the invitation, and hello to you, hello to the listeners.
Exactly, hello to the listeners, and today a guest who is a postdoc and who is working on a very, very timely topic currently. So I was thinking maybe we can start with that. A big story really, and something that I could say relates to all of us. So I would like to find out more about that. What I already know is that you have been working on the spike protein of coronavirus. And let me try to put it maybe in a bit more context. The spike proteins are the surface proteins that are responsible for targeting the cells and also entry of the virus to the cell, right?
Yes, basically the virus-host interactions.
Exactly. And this is also the main focus of the vaccination development and the efforts that are made in this direction. You were developing simulations for the modeling. So can you maybe explain what exactly were you looking for and how did you get involved in this?
Yeah, this was a very, very romantic story, actually, how this whole thing started. So before I've been working on the simulations of the cell adhesion proteins, which are in the membrane. They're mediating interactions between the cells. And I've been working on that for a long time. I created a big simulation model. I could explain the adhesion between the cells. And then when the pandemic started, my first thought was, okay, so the virus has to interact with the cells, and how virus-cell interaction can be different from the cell-cell interaction. It's going to be that hard. We started very early. We started already, I think, in February. We had the first discussion that it would be great to assemble a special task force at the Max Planck Institute of Biophysics in Frankfurt, where we gathered specialists in different subdisciplines of the nuclear simulations. And we were just brainstorming what we can do and how we can help in this situation. And we had people that are great in the membrane simulations, big protein simulations, protein-protein interactions. So this was all like a very good team that could tackle the spike protein. And as you said, the spike protein is mediating the entry to the whole cell. So anything that targets the spike protein is actually great, because we can interact with this, we can block it, then we can block the infection, right? So this was the very, very beginning of the story.
you were trying to understand what's the structure of the spike protein, and I think what you found out is that it's actually very dynamic, right?
This first structure of the protein, because for the simulations you need to have some kind of atomistic structure of the protein that you want to study. And this, we were lucky because the spike protein has been crystallized, and there were also cryo-EM structures very early. Already in February, there were few structures, which were, however, only targeting the very tip of the spike protein, which was easy to express. So the groups that were studying it, they basically cleaved off everything else. The structure of the head of the spike was there, and you have to imagine that the spike protein is like a lollipop. It has a big, bulky head, and it's somehow attached to the viral membrane. So this lollipop per se was known, but how it's connected to the membrane was absolutely unclear. And we decided to model that, and we used some bioinformatic predictions to model how this is anchored to the membrane and what are the sections which are basically connecting this lollipop to the membrane. I mean, the predictions were clearly showing flexible joints between very stiff elements. And that's why we coined the names, the leg of the spike, and we call it hip, knee, and ankle, the joints that we found in the simulations. And we got a big grant on the Supercomputing Center, SuperMOOC in Munich. And we could use 20 millions of core hours to actually do the simulations, because the systems we are talking about are huge. We had around 4 million atoms, which is upper class simulation. It's quite big. And we needed to simulate it for a very long time. And this required considerable computational resources. And the story is very convergent, because meanwhile, the Beck group at EMBL at the time obtained the very high quality cryo-electron tomography images of the whole viruses, whole virions. And they were trying to do the subtomal averaging to get the high resolution structure of that, but they were puzzled by this huge mobility of the connector, which they couldn't solve, because this was always mobile, and averaging was leading to completely blurred structures they couldn't resolve. And they contacted us, and at this point we started working together, and we could immediately explain why they cannot see this leg, because every copy of the spike had a different angle between the joints. And this was the whole issue. And we managed to put a very nice story together. And
So what does this flexibility mean actually for the infectivity of the virus?
Yeah, so we have some ideas why the virus wants to have this leg flexible, because if you look at the whole class of the viruses which are similar to the SARS-CoV-2, and the leg structure is very conserved, which means that for some reason they prefer to have these sections which are stiff and then joints between them. And our speculation is that if you imagine that the round virus, which is rather tiny, is approaching the cell, which is several micrometers in diameter. So for the virus is like a flat football pitch. And if you would have one receptor on the cell, and you would have high curvature virus surface, you could only interact with this one protein, because otherwise the rest would be just pointing outside of your spike proteins. And if you can bend it, so you can retract some of the spikes and then protrude more of some of the spikes, you can have multiple interactions. So in that sense, you can increase the avidity of the virus by attaching to multiple ACE2 receptors. Whether this is the case, we are still waiting for somebody to confirm it. And we're actually working on some simulations that could help with that. But we don't know for sure.
I see. And I was wondering if your work, if your simulations can actually help with the emergence of the new mutations. A lot of them are actually localized within the spike sequence. Can your simulations help with understanding how these mutations work, or with designing new strategies?
I mean, definitely designing new strategies is something we've been already discussing for a while, how we could answer to the new mutations. And I think it becomes clear that the escape mutation is only a matter of time, and we will have a new wave of the virus which is not responding to the vaccines, which is pretty bad. So it's very important to be prepared for it. The simulations can help. You could actually rerun the whole simulations and try to understand if the certain mutation is affecting stability of the spike, how it's exposed, and so on. The more practical way to look at it is to look for the potential epitopes that we can use to target the spike protein in the vaccines or in the antibody treatments. Because the mutations will be affecting the affinity to the ACE2 receptor, the main partner of interaction of the spike protein, but they are not affecting so much the exposure of the pieces of the spike protein.
Yeah.
And so if we find a good strategy that we can use to target parts of the virus which are not susceptible to change, because some parts are very conserved, then we could actually end up with the vaccine which is resistant to the escape mutations. Now, at this point, I have to make a small detour and tell you about the glycosylation. The spike protein is not only the protein, but it's also covered with the sugars. The sugars are called glycans.
Yeah.
And the spike proteins of the virus is actually hijacking the human machinery of the post-translational modification of the protein and is covering its surface with very big branched sugars, which have a main role in preventing the interactions with the human immune system. So they cannot be too immunogenic, because otherwise we would be autoimmune all the time, because we are also using these glycans to recover our proteins. And this is a very good strategy by the virus, because if it's covered, then it becomes basically invisible to the immune system.
So it's like a shield.
It's like a shield. Okay. Yes. Yeah. Yes. And you can imagine that this shield is very mobile. So it's not like one wire of the sugars, sticking out one direction and the other one the other direction. The sugars are basically moving like sweepers on the windshield in your car. At any given moment, you only see one line, but essentially, if you average over time, they are very effective in cleaning the surface. They are everywhere. Yeah. And this is the same with the sugars. They are sweeping the surface of the virus all the time. And this makes the binding of the antibodies very difficult, because at any given time there's some sugar interfering with the binding. And the idea is to find places which are not very effectively shielded by the glycans, and find them in positions which are not close to this domain which is used by the virus to bind to the ACE2 receptor. Because this domain, surprisingly as it sounds, is actually the most variable. So there is some motif which is binding the ACE2 receptor, but the rest of this domain acts like a decoy. So it's actually attracting the antibodies because it's very exposed, and it has very flexible loops which are easy to bind, but can also change very much. So once the body starts producing the antibodies against this decoy part of the virus, then the new variant comes, which is changing a little bit. It again becomes invisible because the antibodies don't bind there anymore. So the whole trick would be to find space on the surface of the spike, which is far away and is perhaps important for the virus when it's performing this strange maneuver of transitioning to the post-fusion state, which is mediating the connection between the virion and the host cell. If we can block that, then the virus cannot enter, and it can bind, but it will be swept by the immune system. So this is the whole idea. And we are now working on the simulations to predict what are the good places for that. We actually already published a paper about this. And we found several epitopes which could be used potentially by the companies which are producing the vaccines to produce a new generation of the vaccines.
And this method, this approach that you're proposing, it can actually be applied to many other viruses, right?
Actually, it could be applied to any pathogens, basically, right? Because if you have a structure of this protein which you are interested in, and you have some idea from mass spectroscopy, for instance, about the glycans, you can start to model this, and you can predict where the unshielded places on the surface, and then maybe devise the constructs for the vaccines which are only presenting this part, right? Because we don't want, as I said, we don't want to target the super immunogenic decoy sequences. We actually want to cover them, but we want to present something which is more conserved, but maybe not as exposed.
Yeah. This is amazing. I mean, it's extremely important work, and if it helps with getting even more efficient vaccines, it's incredible. And you've been actually doing this while you are affiliated with two different institutes, right? You are with Max Planck and also with the University of Vienna.
Yes.
So how challenging has it been to work in like two institutes and so many collaborations? Is it actually very hard, or the fact that you are anyway currently because of pandemic stuck at home didn't really make so much difference?
I mean, the fact that I was sitting in this very office for a year, it didn't make that much of a difference in terms of working. Before that, when we still could travel normally, it was, of course, a bit challenging because then I was basically 50-50 sharing my time between Frankfurt and Vienna and traveling every 2 weeks. But it was also a good time. Like changing the environment, I think is very good for the thinking, and also working in the train is actually great.
So how long is the train ride from?
It's 6-ish hours, and you have very good internet, and you have no administrative tasks. Nobody's calling you. You can just sit down and do stuff. It's actually very good.
Okay, I'm gonna have to believe you on that. I'm not sure if I would do so well if I had to work from the train. So, okay, I think we covered your most recent work, and now I would like to jump a bit in time and go back to much, much earlier days. Do you remember at what point you started being interested in science? Was it physics? Was it biology?
Actually, the very, very beginning, I was interested in the Polish philology.
I was interested in law, so, we all have to start somewhere.
Yeah, so I think this was more like a family business because my dad is a professor at the university in Krakow, and he's a dialectologist. And when I was young, I was like going with the students for the field work, and I liked it. Dialectology was great, and that was initially the idea that I could do that. But then, being rebellious when you are a teenager or something, but then I switched completely to the hardcore science, basically to physics, biophysics, and got hooked up by the simulations at some point. When I was starting the studies, there was a new course offered at the university in Krakow where I did my master's, which was completely new. So they created biophysics as a subject from scratch. And it sounded very interesting, very, very new, and connecting a bit of biology and physics. So I tried that. And also because it was a first year, they didn't really know how to give this course and how to do it. So we were attending the chemistry with chemists, mathematics with mathematicians, and physics with physicists. Okay. It was hardcore. It was really hardcore, and it was pretty intense. But I think it was good because it gave us a very good overview and maybe a bit deeper understanding of how these things work. When I discovered molecular simulations, that I was completely hooked up. That was it. I really liked that.
Your work during your master's, and I think also your PhD that you did in Warsaw at the Institute of Physics of Polish Academy of Sciences, was it mainly theoretical? Was it entirely theoretical, just focus on the simulations?
So no, the master's, my initial idea was to connect experiments and simulations, because it's great to do simulations, great to do theory, but it's nice if you can sort of do the check if it's real, because sometimes you can really get carried away with all the models, and it's good to attach them to something firm. So I did my master's, also joint master between experimental and theoretical groups. And we've been working on the high pressure crystallography. So the proteins, when you subject them to the high pressure, they partially unfold. And this is reversible, and you can actually peek into the folding process. You can look at the last stages of the folding process. And you have like a diamond anvil. Between the anvil, you put the crystal of the protein, you squeeze it with the screws until you get some crazy pressures. And you can solve the structure by the X-ray diffraction. You can actually also simulate proteins under high pressure, and so I was trying to do that. It was challenging. I think we never even published anything out of that. It was difficult, and maybe that's why I moved on to the pure theory afterwards, which I did in Warsaw with Marek Cieplak at the Academy of Sciences. I think in the next 4 years I was sort of detoxicating from the experiments.
Okay, so you were detoxicating and running a lot of theory simulations, but then you finished your PhD and you went back to wanting to work in a lab. So
I was missing something, I guess.
Yeah, so now, you did your PhD and then you decided you want to stay in academia. Did you actually consider any other career path on the way, or was it clear for you that you want to do a postdoc?
That was very natural. I think the PhD went pretty well, and I thought that, okay, why not try it? It's cool, and it sort of becomes a hobby. So why change it, And this was the motivation, that people can have chores, they can have hobbies, right? And if I can be paid for my hobby, this is awesome.
Yeah, it's the best
I want
deal.
to do that.
So it was clear you want to stay and do postdoc, but then were you looking for a lab-based biological work, or was it just by accident that you found a specific project that really resonated with you?
I think it was. I was actually advised by my PhD supervisor to go to some hardcore theory group and just continue what I was doing. And I think he was quite unhappy that I moved to the experimental biology. But I wanted to try, let's say, to go back to something firm and real and something I can touch. So in the PhD, I was studying the mechanical properties of proteins, so unfolding and how do you pull on the protein, what happens. And when I was visiting the IST Austria, I spoke with Carl Philipp Heisenberg. He had this project that involved actually the mechanosensing and unfolding of the proteins, which are then mediating the interactions between the cells, and the whole question like why the tissue is is staying together, is not falling apart. This got me very interested, and I thought maybe I can use my knowledge of the proteins and try to solve some mysteries on this front. And I think this was the main motivation. And I think I also wanted to do some real experiments again.
You came to IST for your postdoc, and I think you really jumped to a very deep waters, because before you were doing a lot of this like structure-based lab work, and now suddenly you are in the lab that, well, first of all, works on zebrafish, an organism. I don't know if you were actually also using the fish. And then you... Yeah. Exactly. So suddenly you have to work with a model organism, an animal, and you have to do a lot of things that are, out of your scope. Suddenly you are in the field of developmental biology. What has been the most challenging in this transition?
Yeah, I guess the first year was just understanding what they are talking about. This was like, I didn't understand a thing. I mean, maybe not a thing, but it was very difficult to follow group meetings. And I think this was hard, but this is like learning a bit the vocabulary, and then you connect this vocabulary to things that you do in the lab, and you see, and it's like, okay, start to understand. And also, I think the kitchen, so that working with chemicals and RNA synthesis, all these things, they were quite manual, and I think I had to learn quite a bit. And the pipettes, I mean, this is what I was doing. It was a dual pipette assay. So you have like two microcapillaries, and you can control the pressure, and you can basically grab the cells by these capillaries and put them together, and then try to detach them to see what is the force. But this is per se extremely fragile experiment.
High precision work. My God.
Yeah. So I was joking at the end of the postdoc, because I became like good with the pipettes, but I became a master of the skill that I will never ever use again.
Was it a difficult thing for you to work experimentally? Because, of course, in experimental work, so many things can fail. And like so many things can go badly, even not by your mistake, but just, the fish don't lay eggs, and something is not working, like exactly the chemical or something like this. And sometimes it takes several days to get to the point where you can actually get the result, and then it doesn't work. Was it something that was a problem for you? Was it a new thing for you?
This was eye-opening, I would say, to like how much maybe I'm saying something wrong here, but how much the results we are getting or how much performing of the experiment depends on your own machine learning network, that you get the feeling for the things. Like if the cells look good on that day, if they look bad, sometimes you cannot even say why they look bad, but you it's
have
not
the
feeling. You just have the feeling, and this was very eye-opening for me, that it was like, okay, so we cannot control everything, unlike in the simulations where you can have everything is deterministic. You can go back in time, you can go forward in time, you can do everything you want. And here you don't. And you have too many parameters that you cannot control. And this was for sure interesting experience. And also the linearity of making the experiments. If you make a mistake anyway on the preparation steps, the whole day is gone. Right? So you can start at 7 a.m. and then at 3 p.m. you are starting on the microscope and you mess up something, some small detail, and okay, over. I go for coffee. That's it. So this is different.
So I think it's safe to say that, you gain a bit of appreciation, because I guess in your simulations you were also using experimental data, but sometimes you don't realize how much work goes into it. And
work, but also like I'm always saying that this is I finally understand what are the error bars, because you get some number, and you maybe trust it, you maybe don't trust it, but what it really means, what is the variability, why this force is like that. I think this was very important to understanding, and also I think it weighted heavily on my next steps. That now I'm using the experimental data. I'm actually trying to even open the raw data myself and look there and see how they did it, and not just trusting that this is how it is, because sometimes the details will matter a lot. And I wanted to add that in the simulations you have some scripts that you are preparing, and you can always have a copy of the script, and if it fails you go back and you just debug it and you run again.
It would be much faster if we had this chance with our experimental work, I would say. So but now, as you said, you don't do any experimental work, you're just doing the simulations. Do you actually think it may change again?
Who knows?
So you see the possibility.
Yes, but the thing is that I stopped doing like theory per se. I'm only doing like very collaborative projects with experimentalists. I think I'm trying to at least talk to them and understand how they are doing it, see the experiment, and so on, to get a good feeling how we can simulate the same conditions, how we can connect the things. So I think this changed forever in me. I will try to do that. Whether I will do the experiments, I don't know. Maybe some small things.
But I think this is good because you got the perspective, and there's not so many of the biophysicists that actually tackle the experimental part themselves, and it gives you a bit of a unique view on the collaboration with the biologists. Is it important for you to collaborate now more with the experimentalists because you want to validate your simulations and kind of have a way of knowing if it works? Or is it more about the translational aspect and the applicability of your work?
I think it's a very interesting question. I think very personally, I feel that I feel it more rewarding if I can check that my simulation is valid. So the validation aspect is great. And also that I can actually help, other group in understanding. So I think this merging and going like quantitative structural biology aspect of it, I think this is great, and this has been like driving me for last years. I guess this is my answer to that.
So what are your plans now? Do you plan to remain within the academia, and do you want to have your own group?
Planning is one thing, and reality is another thing.
Of course, sure, but like,
if you ask what I want to, yes, I would like to stay in the academia, and I would like to start a group and hopefully connect the experiment with the theory under one roof.
That would be cool.
This would be my goal. Whether it happens, I don't know, because the funnel is narrow, and
sure.
I did the second postdoc, and also have to be realistic about that. So that at some point it becomes very difficult to stay. I don't know. I'm definitely fighting.
It is harder as the time goes, but on the other hand, I think you are really doing a completely different thing now. So this gives you a bit, better expertise to start the group.
I mean, the accusation sometimes is that I am too broad by the reviewing committees.
I mean, you have a very different projects. I mean, I can understand, but on the other hand, I think this should be viewed more as a. It's kind of cool that still from all of these projects you can just take what you learn in the previous group and apply it to another group, and it's still useful in a completely different branch. I mean, you just worked on the development, now you're working on again the structures, and you're working a bit more in the translational aspect. I think it's cool that your work is so applicable actually to so many different projects.
Mm-hmm. Not too many places can host such a group because they are either physics or maybe biophysics, or they are experimental biology. And this has been, I think, the difficult part, that they don't know what to do with such a person.
I can understand that, but I really hope you can find a place. And now there's so much talk about interdisciplinarity. Institutes are slowly moving into this direction, but still, as you said, they kind of hope that there will be collaborations between groups, and there's not so many interdisciplinary groups per se. It's a bit of a risk, of course, for the institute, but I think, I really hope you can get a place.
I mean, the whole science is a risk. Scientific career is a big risk.
Exactly. So what do you think one should really work on if they want to go towards becoming a PI, except for the research work? Like from your perspective now, in retrospect, what would you say is something that maybe one should focus in addition to research, or like pay less attention to, and maybe you can give advice?
Yeah, I think that all the soft skills, like negotiation, very good writing, fast writing of the grants, fast writing of papers, and the writing per se, I think this is the essential skill of everybody that wants to stay in science. And I have a feeling this is not emphasized enough. That you have to train. You have to train it. You have to do it a lot. Also, learn how to fail and how to learn from these failures. How to become again faster the next iteration and so on. I think this is crucial. What else I could say? Supervising others and doing it even early. I think this is also something which gives you a whole new perspective, which will be then useful in the future when you actually become a PI. And there are parts which I personally don't like, but I think they're also important because, for instance, I don't feel very well when I'm teaching. I'm more like a one-to-one person. I can explain things, but I don't like talking ex cathedra and explaining things to the crowd. Yeah. And this is sometimes those fears have to be overcome. And also, I think this is important to jump into the water and also do these things. Yeah, but there are many soft skills that you need, and you only learn when you are starting to apply for the PA position. How many skills you need to know? You need to be good in the buying computers, because you need to explain to the granting agency why you need this cluster and not the other. You have to be very good in your normal trades, of course, that you are doing, like your software that you want to use and so on. This is crucial. You need to be able to negotiate effectively. Yeah. You need to be able to convince people to present in a very good way. So there are like countless skills. Also, solving the conflict and working in a sometimes hostile environment. Let's be honest. This also happens in science, that the environment is not so friendly, and you have to be sort of tough to sort of survive it.
Yeah, I think this is important, and I'd like to maybe touch on another thing here, which is the networking. I think this is something that maybe at the beginning of our careers we don't realize that it can really change a lot and open a lot of doors. And I'd like to also discuss a bit your involvement in this Polonium Foundation with respect to that, because this is an organization that is unifying the Polish expats, right, in science. And you have been involved in this organization, right? So what drove you towards getting involved, and what experiences did you get through your work with them that are actually helpful for your career?
I felt that this organization is doing a great job, because many things that you cannot learn by going somewhere and asking somebody that you don't know anybody. It's much easier if you know some compatriot, and then you can access him very informally and ask. And it seems that this channel is giving you much more honest answer, and you can also ask about stupid things like buying bread or buying something you just are used to in some other country, or how to do taxes. All these things are kind of difficult to obtain via the official channels. So I think this is a great concept behind being involved in such a community of expats which are making a global network, and then you can always find somebody that would be helpful. I think I was not as involved as my wife, who was basically preparing this database of the Polish expats, and I was merely helping her with that. So I would not want to claim what is not due, but I think I sort of saw enough of this environment to really enjoy it and like it, and I think this was a great experience to know people from many different countries which are still sharing very similar problems.
This website, this database that you're mentioning, is called Polonium Network, right? And this serves to, just for the people who might be interested in joining it, it basically is supposed to be an easy address book for you to contact someone, maybe at an institute that you are looking into, or a country that you are considering to move, and to exactly have this a bit informal conversation. From your perspective, as already a very advanced scientist, I would say, in comparison to me at least, how much did networking help you?
I mean, I would say it's impossible to go without that. You have to know people, and you have to also show yourself what you are doing, not only do it in the quiet lab and stay there. So I think it's absolutely essential to go to the conferences, if you can go to conferences. And even if you cannot go to conferences, at least go online and show them your face whenever you can, and talk to them, and convince them that what you're doing is cool, and you're a smart person, because then it's as you said, this is opening many doors, opening very many collaborations, because you sort of start to get the brand that what you're doing is solid. And if somebody is then having some problem, like in my perspective, if somebody has some experimental question they cannot solve, they would maybe contact you and ask you if you can help them doing some simulations, and this is crucial. You also never know who would be in the committee when you are getting hired, right? It's brutal, but
it's true.
but it's also really true. Yeah. So I think this was I owe very much to my PhD supervisor, who was sending me and others to as many conferences and meetings as possible. Schools, like 4 times a year, we were going to places and meeting people, and I think this is very important. So for those of you who are becoming PIs, I think do it this way. Let the students go, and not not keep them in the lab all the time, because I think this is also benefiting the PI in the long run, that they know people, they attract others, and other topics and new collaborations for the whole group, new skills. I think this is extremely important.
It's important for the science as well. I think, your recent work is a great example that so many people can collaborate and produce very quickly, actually, so much data. Because we get stuck sometimes in this feeling that in this run for like having higher and higher published data, and there's a lot of competition in science. Sometimes you get it into your head that, everyone can scoop you, and then you will lose your paper, and then you will lose your career. But actually, the collaborations are very important and cannot be put aside. Otherwise, you're really just hurting yourself, because you could get there faster.
I think so, yeah. And I think also we are maybe at the brink of the change because of the pandemic. I have a feeling that the science became much more open in the last year than was ever before. Whatever was published was getting onto the bioarchive immediately, as there was any kind of very preliminary results, very drafty manuscript was already there. Data was being released to the others. And I think this is great. And maybe we can keep at least something positive out of this pandemic. That's, sharing means faster progress, and we can just beat any problem if we try to be very open. And if everybody's trying to be fair, I think we can gain a lot from that.
Yeah, I think people get lost. There's, of course, your own ego that comes into play. And then you have, of course, as you mentioned, the funding bodies that sometimes the interpretation of your list of publications is maybe not keeping the best of science in mind. It can change people's perspective when they feel like this is the goal, instead of thinking about science as the goal and like pushing science forward as a goal.
Yeah, the numbers, the impact factors, right?
Impact factor and all. We should move away from this, I think. Okay, let's go now maybe then into improvement in science. So as we said, this system, it maybe needs a bit of update. And as you said, we should be more collaborative in science. Is there something else that you feel strongly that should be improved to make our lives better or easier, or, to help either scientific environment or the academia to be better?
Mm-hmm. So I think one observation I had at the IST was that I really liked what the staff scientists were doing, and I had the impression that in many cases, the postdocs and PhDs are going through the same problems. They are making same mistakes all over again, and they can be easily avoided. If you are doing sequencing, or you're doing any other experiment which is very repetitive and it just has to be done with high proficiency and by the expert, and then you put a bunch of PhD students which don't know how to do it, they waste time, waste resources, and they do it super slow and make many mistakes.
So they learn through the process. Exactly.
Of course they learn through the process, but I was thinking, observing the staff scientists, wouldn't it be better to professionalize a bit this part, not rely only on the PhD students to perform the repetitive tasks, but to have somebody who is really skilled in that?
Well, it would help with many things. It would help with reproducibility and also save time and money.
Transfer of knowledge within the lab, right? Because it's the stays, exactly. And they can teach many people, but in the same way. So this is, again, reproducible on the lab scale. And I think expanding this world would be a very good thing for us. Also, we always lured when we enter the PhD that everybody would be a PI, but in reality, only like less than 5% has a chance to do this, right? But many people actually enjoy doing science. So for them, this kind of positions, which would give you a opportunity to play around and do your hobby and have a permanent position, without the administrative chores.
Yeah.
I think this is something maybe to consider that could be expanded,
I actually, have not been in so many institutes, but to be honest, I don't think this is so popular yet. I know that there are facilities. This is already a big improvement that, now there are people that can help you with the designated parts of your project and they kind of take care of training and stuff. But I haven't really met so many places that have staff scientists, because this is a person that in general would be kind of hired by the institute, right? It's not a lab member, but kind of a bit more institute-based person that has like a permanent position to support. So what do you think is the biggest problem in getting there?
Yeah, I don't know. I think that maybe there was never initiative to do that because the PIs were sort of carrying it. And I mean, who is carrying is the PhD students and postdocs, let's be honest, right? Who is doing the heavy lifting? The system, I think it's sort of working. So I think there is never initiative to change if things are fine, right? Even if it's not too great, you will not have any revolution, right? And I think that we also haven't reached that point that the students are going on strike and not doing their jobs. So it might be a very slow process to actually get this change to happen. IST actually is going, I think, is doing a good job in that. They have some staff scientists.
Yeah.
There are some staff scientists at the Max Planck, which are very respected and doing really great job. And I think it's important to also give them some freedom to have projects on their own, because this is what dragged them into the academia in the first place. And I think it would be great if they can like share it between working for others, but also working on your stuff, because then you get extra motivated and you can do much more. I think creating such positions, it's taking a risk in a way by the institutes to create such positions, right? And maybe it will change.
Yeah, I mean, there are several different models of running institute that are kind of risky and are functioning out there, right? We have some institutes that try to go by this no application for funding model, so they kind of provide you with the money and you don't have to care about funding, and they are trying to see how the science will move forward with this. And I think, some institutes are trying this format of having the staff scientists, and I think it is good that there are some changes coming, and I think what you're pointing out is very cool, but it really requires the PIs also to trust this system more.
Yes, for sure.
Yeah, and I think this is something that we still have to get there.
Yeah, but the same was with the core facilities, right? That in the beginning, we can see it now in Poland that there are fights over the core facilities.
It's starting.
Yeah, and there are some small wars because people don't want to give up their own precious tool that they acquired some time ago, and they don't want to have it shared. But this already happened in the West. Yeah.
Yeah, there is a problem of sharing, with people having to pay for the space in the lab to the university, and then they have to buy the equipment by themselves, and somehow they don't want to share because the money is sparse. But it would be good to see these changes in Poland as well, like more on a regular basis, not just in a few institutes.
Yeah, I mean, there are some places which are good, right? And I guess this is leading by example. And if you see that this generates the net gain,
the results,
then you are much more likely to invest in this kind of thing and risk it, right? Because let's be honest, you are risking something because you are giving up your resources.
Exactly.
And whether you get back what you gave, this is a risk.
As you said, science is a risky business, and it's always about this, like taking the risks and weighing them and deciding into what they want to try to go. So let's move a bit away from science. And one thing that I think we all struggle with is the life-work balance. And I think...
What?
Yeah, exactly. But, let's be idealistic for a second and let's try to think about it like it exists. I think it makes you enjoy your work more when you have actually outside of academia a bit of a relax or a bit of a break. And it can help you being successful, but somehow in our minds, and also there's a bit of a culture that makes you think that it will just make it harder for you to move higher on the ladder. So I was wondering, how do you see this? How do you keep track of, of yourself, and how do you make sure that there is some balance? Especially now, because, we either go to work, at least experimentalist, and then we go home, or we work at home, which is even more difficult to separate these two. How do you deal with it?
Yeah, so I call it the tripod model of the work-life balance. So I'm always saying that it has to be like in some sort of a harmony, and some sort of it requires some sort of caring about that, is to have a family things right, to have work things right, and to have some hobby. Mm-hmm. And if these three things work, then you actually can say you are happy, and it gives you some feeling of being fulfilled and enjoying what you're doing, right? And if any of these things starts to malfunction, then I have a feeling that the others are suffering very quickly too. Mm-hmm. So I'm trying really hard to have this kind of balance. Maybe it's not easy to do it. The sharing of the time is probably not fair. I guess the work is still getting a bit too much from our lives.
Yeah.
but this is how it is. But at least with what's left, I think it's good to make a dedicated effort to have the other parts, good, devoting some time for the relationship, for the family, and then having your own thing that it's only yours, and you can just close your. I mean, at least for me, it works like that. I'm sort of introvert person, and I like to have my own hobby, which I can only be on my own, and get rid of all other distractions, forget about the stress, and then I can feel my batteries charging. Right. So this is how I'm trying.
So what do you do in your me time, for lack of a better term?
In my me time, I'm an outdoorsy person, so I'm trying to stay, either go to the mountains for hiking, or I try to go climbing, and this is my thing. Not the team sports, but rather individual sports, and competing with myself,
challenging yourself. No, this is important what you're saying. This balance is hard. There is a strong feeling, and also sometimes it's not even a feeling, but we are being told that you have to put more and more work, otherwise you will not succeed. And this is really one of the things that I think should be, the quicker you realize it, the better, that it's not really true. Because if you push yourself too much, as you said, the balance is shifted and you don't enjoy any part of your life anymore. And the work, I think we all do this because we really enjoy science, right? But if you push too much, you also lose this. You lose the spark. And it's important to keep this in mind.
Yeah, I mean, this is experiment for everybody to do, is to do what you really like, devote one day, just do it, and then try to solve some problem that you had at work on the next day. And it just goes like that. Like no problem, right? And then you try to sit for a week straight and not go anywhere except for eating and peeing and just sit. And then the last day, it's difficult to draw a straight line on the screen. And I think this shows you that it's really important to also get like discharge from this.
I think there is a lot of internal pressure that we put on ourselves, and especially when things are not working, you have this very counterintuitive feeling that if you put more work, maybe it will work. And there is this guilt maybe that you should have already solved it and you didn't. And when you hear someone telling you like you should actually take a break and it will work, you don't feel like it's gonna work. Exactly, but I hear this over and over, and I actually tried this myself and I was surprised. And I think this is a very valuable lesson that you mostly learn it during like your more independent work, like PhD or postdoc. It's important to keep this in mind, that a break can really solve this, not more work.
Yeah. And also, like I had my best ideas always when I was like physically tired and my brain started wandering around and just, I don't know, after a long hike, legs are heavy, you have so many kilometers in your legs, you're going down and you're like, oh, wow, why didn't I think about it before? And these are this eureka moments for me that actually happen when I'm tired and my conscious brain is not there anymore. And I'm like just
shifting,
thinking about stuff, right? And then the good ideas come.
Yeah, I agree. But as I said, it's a lesson that we all have to learn through experience. And I hope the more we talk about it, the more people will try to believe it and follow it through. Okay, so let's get to the last part of the interview, which is the cool science and the cool people in science. So one thing that I like to ask my guests is if you could work on anything in the world, and I think with you it could be a discussion because you have very broad interest, as you said. What would you like to work on? What is something that is very interesting for you? And if you could, there is no money limitations, you could just go somewhere and do this, what would it be?
My dream would be to be able to have a model system that I could clone in the computer. I could have exactly the same system experimentally and computationally.
Okay.
And then this is opening the whole new field, because you can do the debugging on both ends, right? Something is not working, then you can debug the experiment, or you can debug the model.
This would be really cool.
I'm really hoping that one day it will happen, that you will be able to start very small and start, part of the membrane with one protein in it, and work on both ends until you really converge on everything, really understand this part, right? You just know what's happening there. Then you can modify and you can complicate and so on.
There are some initiatives about modeling the cell behavior, right? That are international. Yes. So it's going to this direction. It would be really cool. I mean, organism is like way, 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 way ahead of it.
Yeah, but this is right?
This is a cool idea and cool science, really. I agree. So the other thing I like to ask is, if you could have a chance to talk to someone that is either currently living or has already passed away, a scientist or someone who inspires you, who would you like to talk to?
I have to say that I was very lucky in my career. I met really great guys, and I have my mentors around in a way. So I don't have this kind of craving that there was somebody in the past that would open the door for me. I think we should appreciate more the people that we have around, and maybe not dream about some unachievable conversations, but just try to focus on what we have and try to extract the best sides of these people that we have around, because everybody has, of course, some issues, but also there are so great people with great approach to life, great approach to science. And it's sometimes just look at it from this perspective that, okay, this guy is really good at that. Can I maybe, if not copy, but at least learn something from him, like how he's doing that, how he's managing the people in the group, or how he's managing the network, or these things. I think this is, we are not appreciating it enough. That sometimes it's enough to just look around. Have your mentors everywhere around you if you want.
So do you think in the current age, because of the online availability of people, it's actually easier to reach out to someone that you kind of look up to, and or do you think it still can be tricky? How do you see this?
I think, unfortunately, in the pandemic, it's a bit harder to establish new relationships. If you know somebody already, I think it made it maybe even easier to communicate and push the projects together and talk about science and discuss stuff. I think the barrier has been lowered, and it's a bit easier to start, Zoom, and then you immediately have an answer. Or I don't know, I have many people on WhatsApp, which we are just talking, exchanging screenshots, like, okay, this makes sense, doesn't make sense, yes, no, and this really is great. But I have a feeling it's difficult to meet somebody complete stranger and just strike a conversation and then start a new project. So there are two sides to it.
I agree. All right. I think we have it all. So I would like to thank you very much for joining me on the podcast and for sharing your story. And I really appreciate it. And I wish you all the best for all your future projects and all your plans. And thanks.
Thank you so much for having me.